Swayam Prabha. Digital India. Educated India.
So, welcome back to the lecture series in animal physiology. So, today we are into the third lecture. We are into week 8, lecture 3. That is where we are. So, at the neuromuscular junction, if you look at it, how the neuromuscular junction really looks like. Okay. So, we have already talked about the signal originating from the muscle spindle going to the spinal cord. They are bifurcation of the signal into the alpha and the gamma motor neuron, and gamma neuron, motor neurons coming back and synapsing on the muscle spindle, whereas the alpha are synapsing on the extrafusal muscle to bring back their length. So, now this is the backdrop of the story. So, now let us see what is happening at this site. At this site, if you blow it up, you will observe neuronal terminal is something like this, like fingers like projection all over the place like this. In a microscopic detail, this is which was observed by the scientist very early, something like this. And these were all the very early electron microscopic picture during 1950s and 60s. Likewise, this is one of the synapse which has been explored by neuroscientists very extensively. We know a whole lot more about these synapses than any other synapse. So, this is where it is sitting. Okay. Okay. Now, if you see these terminals very carefully, you will observe something very interesting, that they have these kind of buttons like structures, and there is a huge amount of mitochondria which are located in these locations. Okay. Okay. So, now this is how it's physically, if you have a very good quality biological EM, electron microscope, this is what you are going to see. Now, suppose a signal arrives here and train of action potential arrives here. As the train of action potential arrives here, these neurons, these ends of this neuron, what these you see, these are synaptic vesicle. These synaptic vesicles are filled with neurotransmitters. Okay. And most of these neurotransmitters in the mammalian systems is acetylcholine. And because these synapses are governed by acetylcholine, these are also called cholinergic neurons and cholinergic synapses. Cholinergic neurons or cholinergic synapses. Okay. Now, at the cholinergic synapses, when the electrical signal reaches here, these neurotransmitters are secreted at this synaptic space. This is the synaptic space, and if you wanted to see it, it will be more like, I think this is good enough. Okay. These acetylcholine molecules goes and binds to the muscle. Okay. These acetylcholine binds to, let me write it down, acetylcholine molecules binds to the muscle. That opens up non-specific cation channels in the muscle. Okay. So, just if you go back a little bit, backward, so there is an action potential, at cholinergic synapse. Okay. These acetylcholine molecules then are secreted at the cholinergic synapse. They bind to the muscle. Non-specific cation channels are opened in the muscle. Fair enough? Now, at this spot where non-specific cation channels are open in the muscle, there is an influx of sodium ions into the muscle. This influx of sodium ions into the muscle leads to the generation of action potential by the muscle. Okay. And in that process, muscle generates its own single action potential. So, there is an action potential which is coming from neuron, and there is an action potential which is generated by the muscle, and there is a role to that, how that action potential which is generated by the muscle is transmitted. So, what you see that there is a wave pulse, one second, on the muscle, there is an electrical activity which is spreading out. Okay. So, now what we haven't talked about is something else. End of the day, the muscle has to perform an electrical function, not only an electrical function, a mechanical action. How this mechanical action happens? So, now having gone this far, you have to realize one very interesting aspect. Before I get into that aspect, muscle is a three-dimensional structure. If you look at a muscle, it is something like this. It is a structure which is like this. Like this. Okay. Now, your synapses are happening at the upper surfaces. So, synapses are happening here. So, electrical signals coming like this, an electrical signal generated by this muscle. Now, think of a situation before I get into the details of it, how the electrical signal could travel all the way into this, or how the contraction can happen all along this thing, because your connection is happening exclusively out here. So, how it is getting transmitted? So, that is where comes the significance of the action potential which is generated by the muscle. And second thing, if you look at the skeletal muscle structure, this structure is very, very unique. If you look at the structure, you will see something like this. This is from one side I am showing you. If I show it from the other side, it will be something like this. Okay. As if there are, you must have seen gorges, as if the structure is gorged like this on both sides. Okay. Why this structure has been made like this? When I drew this cylindrical structure, I did not show this. Now I'm showing you that, I'm adding up to that. So these tubes are something like this. Something like this. Okay. Why the structure has to be like that even in the first place? Okay. Something like this. Now, this structure has to be like that because it has certain very interesting advantages, what we are going to talk about now. Now, say for example, let's take this situation. So, let's take a simplistic view of it. So, here you have the synaptic zone. So, action potential generated by this. The muscle generates its own action potential, which is shown in blue. These action potential travels like this. Now, out here, there are some very unique features. And I will come to this unique feature, which will sum up the whole story. There you have a lot of calcium sensors or calcium ion sensors. What are the calcium ion sensors? So, there are something called, one of the key ones, calcium ion sensors are, it's called RYR DHPR receptors. We'll talk soon about the locations of this. Let me give you the full form. RY stands for ryanodine, R stands for receptor, D stands for dihydro, P stands for pyridine R stands for receptors. So you have these very two interesting receptors or calcium sensors and calcium channels, whichever we call them, because these are very controversial till this date. Ryanodine receptor and DHPR receptors, and they remain in close proximity like this, okay, something like this. Now what happens is very interesting. Now let me go back. So most of this, now I will use a different color code for you to understand this part, okay. So most of this DHPR, let me take a slightly brighter color, okay, and dihydropyridine receptors are sitting like this. These light green colors you are seeing, this is where they are sitting, okay. Now this is where they are sitting. Now when the electrical signal comes, so electrical signal can gorge inside these places, okay, because it is a gorge like structure, you could see. So the electrical signal comes here like this in the muscle, it spreads out here. Out here it encompasses these, so now I'm showing in red the DHPR and ryanodine receptors. So what happens, these receptors senses electrical signal, and part of this dihydropyridine and ryanodine, which is still not very clear, is attached to an organelle within these called SR or sarcoplasmic reticulum. Sarcoplasmic reticulum is a storehouse of calcium, okay. Sarcoplasmic reticulum is the house of calcium which is sitting there. Like this. So this is the storehouse of calcium. Now when the electrical signal comes here, these electrical signal makes this sarcoplasmic reticulum sensitive to it. They become getting sensitized because of this adapter kind of molecules, which are the calcium sensors, which are ryanodine and DHPR receptors. Okay, this ryanodine and DHPR receptors lets this sarcoplasmic reticulum to give out the calcium. This calcium now initiates what we talked about is contraction process in the actin myosin filaments. So that whole movement on the actin myosin filament is executed by the calcium which is secreted at that spot. Okay. So this leads to what we call as sliding filament motion of the muscle. If you remember, when we talked about muscle, we talked about this sliding filament motion of the muscle, where this sliding happens, and this calcium spike comes from here. This is the calcium spike which is coming. So it's a very interesting process how these electrical signals are transmitted at the 3 dimension. back and forth. And which leads to the mechanical motion of this muscle. So this whole process is called excitation, contraction, coupling apparatus, because there is an excitation which is electrical excitation, which I told you what it is, leading to an contraction which is a mechanical contraction, electrical excitation leading to mechanical contraction. So, excitation, contraction, next thing what we know is coupling of these two, and this whole process is called excitation, contraction, coupling apparatus, which contraction is because of sliding filament motion of actin and myosin filaments, whereas electrical excitation comes at two levels. One is an action potential from alpha motor neuron. Second level is, if this is the first one, second one is action potential generated by skeletal muscle, and this action potential generated by the skeletal muscle leads to a muscular contraction governed by sliding filament motion of actin and myosin. So, this whole thing is very wonderfully coordinated at different points. So, the first electrical stimulus which is happening at NMJ or neuro muscular junction, the first one. Second, this muscular contraction is happening at this zone, is happening of course on the muscle, but this translation is taking at RYR DHPR interface. So, we see at different level these information transfers are being governed. Okay. So, that's what we needed to talk about the neuromuscular junction and the stretch reflex circuits. Now, from here we will shift the gear and we will move towards the special senses. So, I will close in here, and thanks for your patient listening.